Welcome to The Explainer. Today, we're looking at what might literally be the ultimate construction project. Imagine you've got the master blueprint to a house, right? And with just a few strokes of a pen, you could, I don't know, add a whole new wing, change the color of the bricks, or make it entirely stormproof. Well, nature has a master blueprint too, and we call it DNA. In this explainer, we're going to dive into how science is learning to read, edit, and entirely rewrite that biological blueprint through genetic engineering. So what are we actually talking about here? Genetic engineering isn't just some far-off sci-fi concept anymore. It is the very real, deliberate modification of an organism's DNA. We're using advanced molecular biotechnology to introduce, remove, or modify specific genes. You see, we aren't just crossing our fingers and hoping for a lucky mutation anymore. We're intentionally altering characteristics to achieve a highly specific purpose. And honestly, it fundamentally changes how we interact with biology. Here's our roadmap for today. We'll start with the blueprint of life, unpack the genetic toolkit, walk through the step-by-step construction process, look at some real-world applications, and finally, tackle the ethics and the future of it all. Part 1, the blueprint of life, a brief history. Because, before you can architect life, you've got to actually discover the blueprint. The sheer speed of human innovation here is just staggering. We start back in 1953, when Watson and Crick first discovered the DNA double helix structure. And remember, that was just discovering the shape. Fast forward a mere 20 years to 1973, and Cohen and Boyer are already creating the very first recombinant DNA molecule. They were literally combining genetic material from entirely different sources. Less than a decade after that, in 1982, the first genetically engineered human insulin gets approved for medical use. And then, the absolute game changer in 2012, the development of CRISPR-Cas9. In less than 60 years, we went from merely observing the blueprint to holding a hyper-precise pencil to rewrite it. Moving on to part 2, the genetic toolkit: scissors, glue, and vehicles. If we're treating DNA like a construction site, we obviously need some specialized equipment. First up, we need to make some cuts. Enter restriction enzymes. Think of these as your molecular scissors. Their whole job is to basically scan the DNA and make precise cuts at very specific nucleotide sequences. Without these guys, we wouldn't even be able to isolate the genes we want to work with. But once you've cut a piece of DNA out, how do you actually put the new one in? Well, that's where DNA ligase comes into play. If restriction enzymes are the scissors, DNA ligase is the molecular glue. It's an enzyme specifically designed to seamlessly paste and join those new DNA fragments perfectly into place. Okay, so now you've got your modified gene, but you still need to get it onto the job site, right? Inside the host cell. We use vectors for this, and a super common one is the plasmid. Plasmids are circular DNA molecules that essentially act like tiny delivery trucks. Oh, and fun fact, scientists can also use modified viruses or bacteriophages as vectors too. They safely carry that new, modified genetic information right through the cellular membranes and directly into the host. And that brings us to our most recent, totally revolutionary tool, CRISPR-Cas9. If the previous tools were standard hand tools, CRISPR is like a highly advanced programmable multi-tool. It allows for insanely precise, targeted modification of DNA sequences. It's faster, it's cheaper, and it's way more accurate than anything we've had before. It has absolutely transformed the entire field of gene editing. Part 3, step-by-step construction: architecting a gene. Let's see how all these tools actually come together on a biological job site. Let's walk through the logical flow here. First up, scientists have to identify the exact gene responsible for the desired trait, and then isolate it from the donor organism. Step 2, we bring out our molecular scissors, those restriction enzymes to cut the DNA. Step 3, we form what's called recombinant DNA. This is where we use our molecular glue, DNA ligase, to join our target gene to a cloning vector, like our delivery truck plasmid. Step 4 is gene transfer, where those trucks drop the recombinant DNA into the host cell using methods like transformation or microinjection. And finally, step 5, selection and expression. We figure out which cells actually took up the new DNA, and we just watch as the host organism expresses the brand new trait. Part 4, real-world applications. The finished skyscrapers? We've seen the underlying process, but what does the finished construction actually look like? It turns out genetic engineering is this incredible multi-tool solving massive global challenges across basically every sector. In medicine, we're producing life-saving vaccines and gene therapies for inherited diseases.
Over in agriculture, we are designing pest-resistant and biofortified crops to help fight global famine. Industry is using it to create sustainable biofuels and bioplastics to reduce our reliance on fossil fuels. And for the environment, we are literally engineering bacteria that can consume waste and assist in bioremediation and pollution control. It's wild.
Let's actually pause for a second on a specific year, 1982. This was the year that first genetically engineered human insulin was approved. To put that into perspective, before this, diabetics had to rely on insulin extracted from the pancreases of pigs and cows. That could cause allergic reactions and was really difficult to produce in huge quantities. By engineering bacteria to just produce pure human insulin, we didn't just improve a manufacturing process, we fundamentally revolutionized modern medicine and saved countless lives. Which brings us to part 5, ethics and the future. The zoning board, if you will. Because with this kind of godlike power comes immense responsibility. I want you to really think about this for a second. We now have the technological capability to alter the very foundations of living organisms. But, just because we can build something, does it mean we always should? Where exactly do we draw the line between a beneficial cure and crossing a serious ethical boundary?
We're facing a really stark balancing act. On one hand, the advantages are just miraculous. We can improve crop yields to feed a growing planet, produce life-saving medicines, and accelerate vital scientific research, all while reducing production costs. But we absolutely have to weigh that against some very real limitations. There are profound ethical concerns. There are potential unforeseen environmental impacts. There's the persistent risk of unintended genetic changes, mutations that we might not be able to put back in the box once they're released into the wild. And, of course, there are high research and development costs alongside some massive regulatory hurdles.
And this leads directly into the specific ethical considerations society has to grapple with right now. Take human germline editing, for instance. Altering DNA that will literally be passed down to future generations. That is a massive, hotly debated topic. We also have to ensure the strict safety of GMOs and vigorously protect our natural biodiversity. Plus, as we map more and more DNA, genetic privacy becomes a huge issue. And crucially, we have to ensure equitable access to this biotechnology, because if only the wealthy can afford these gene therapies, we risk creating a literal biological divide in society. So responsible regulation and intense risk assessment, they aren't just bureaucratic red tape, they're absolutely essential.
But if we can successfully navigate those ethical waters, the future is incredibly exciting. Looking ahead, we're talking about personalized gene therapies tailored to your specific DNA profile. We're developing climate-resilient crops that can actually survive extreme weather. We are even exploring entirely synthetic biology and artificial chromosomes, all of which is being accelerated by AI-assisted genetic engineering. The precision and efficiency of this field are just skyrocketing.
Which leaves us with this final thought. We are no longer just inhabitants of the natural world. We are becoming its architects. As we continue to unlock and edit the blueprint of life, how will we balance our newfound power with our profound responsibility to nature and to each other? Thank you so much for joining me for this explainer, and I really hope it leaves you thinking deeply about the blueprint inside us all.